So today's presentation is for Grand Canyon University's Chem 530 class, the Concepts in Biochemistry. This is a signal transduction presentation titled Receptor Tyrosine Kinases and Cancer by myself, Catherine Conan. So first, before we get into some in-depth conversation about some particular tyrosine kinases, I'm going to go ahead and describe the family of tyrosine kinases themselves, and then we can get into a little bit more depth. So first of all, tyrosine kinases are a family of about 90 different enzymes, and all of those enzymes are involved in phosphorylating the amino acid tyrosine. Now that tyrosine is usually connected to a different enzyme, but the tyrosine kinase actually assists in the addition of phosphate to ATP. Now, in this, once the ATP is phosphorylated, that then can lead to some conformational changes in the enzyme. And that's really, really interesting for this enzyme because this enzyme actually is, part of it is outside of the cell, and it actually spans its way through the membrane, and then part of it is inside the cell. And so those conformational changes based on what's happening outside the cell has an effect on the inside as well. And so that's really interesting. But the protein itself can be activated or deactivated or change the speed with which it performs its function depending upon that phosphorylation. Now, there are two categories of tyrosine kinases. The first one is membrane-bound tyrosine kinases, and that's what we're going to focus on today. This presentation actually is going to hone in on receptor membrane-bound tyrosine kinases, and so we're looking for the ones that are going to take or recognize chemicals outside the cell in the extracellular space and cause a conformational change that's going to affect the interior of the cell and instruct the cell what to do. So we're definitely dealing today with the membrane-bound tyrosine kinases, but there are also other tyrosine kinase enzymes that do work in the intracellular space, both in the cytosol as well as in different membranes of different types of organelles. We're not going to focus on those today as much. But there are some tyrosine kinases that are intracellular as well. So let's talk about in general what a receptor tyrosine kinase looks like and what it does. So if you look at this picture here, you can see that there is the phospholipid bilayer, and above that there is the extracellular domain. Now in that extracellular domain, that is where a ligand is going to come from inside the body and bind that receptor. Those ligands can be multiple different things. It could be hormones, it could be different ions, it could be growth factor, neurotransmitters, different small molecules. We also can try to target that binding site with drugs, but this is where things in the extracellular space are going to bind the enzyme itself, okay? Now, that enzyme, like I said earlier, spans through the entire membrane, and so as you can see, this isn't a pure ribbon drawing here, but it's showing you that it goes through the membrane, and inside that membrane there is actually an alpha helices, a single alpha helices, in which the way that that alpha helices is turned and coiled, the R groups on those amino acids are going to be your nonpolar groups, so that they can sit and be happy and make their way and sit happily in that membrane. Getting into the inside of the cellular space, we can get to the cytoplasmic domain, which is all of this positioning here. There is going to be part of the enzyme that is very, very closely connected to the membrane. And then a little bit further down, you're going to get to the actual meat and potatoes of this enzyme. This is where the tyrosine kinase does all of the action with phosphorylation. Happening here with an N lobe, the C lobe, and in between those is what's known as the activation loop. All of the action for attaching that phosphate group is going to happen in that activation loop, okay? There is also a C-terminal tail, but that kind of gives you an idea of what all of these enzymes look like generally and how an attachment of something in the extracellular domain can affect the conformational change on the inside, and that sends signaling pathways, which is what this topic is about. That's how that signal is going to be passed into the cell. Now, I'm going to focus on a couple different families of the membrane tyrosine kinases. The first family that I'm going to focus on is the epidermal growth factor receptor, okay? This I find actually is the most fascinating of some of them. I just think it's really, really neat how these enzymes work. So the EGFR receptor is involved in signaling growth as well as division for the cell. And so eventually we're going to get into some talk about how that affects cancer growth as well. And so since these guys are involved in growth in general, that will affect, if this cell does become cancerous, that will affect how rapidly it can divide, etc. But before we get to that, what happens here is the epidermal growth factor, okay, is going to attach, again, outside of the cell to the receptor. When that part happens, the next step is there is actually what's called a dimerization step, where two of these enzymes actually can connect to one another. And I think that's absolutely interesting, and that's really something that you see a lot of in this family, is a lot of dimers and trimers, and these proteins are actually binding with one another as part of their normal everyday functioning, which I find absolutely fascinating. So that dimerization actually is what activates the enzyme itself and gets the enzyme to work, okay? If that dimerization is not happening, then we're really blocking this entire enzyme from performing its function and from attaching phosphate groups, okay? Continuing with that, like I said earlier, this becoming a dimer is actually going to cause the N lobe as well as the C lobe to be activated to be able to perform and attach that phosphate group, and that is then in turn what happens next. Now, in some of these enzymes, the attachment of the phosphate groups can happen through what's called autophosphorylation, where they will actually attach phosphates to themselves, but it also can happen in a trans fashion, where this enzyme can attach phosphates for the neighbor, and the neighbor can attach phosphates for the first enzyme. And I think that's absolutely fascinating as well, that the dimer certain ones can pass or attach those phosphates back and forth. Now, once the dimer has occurred in this case, and we've have attached phosphates, the next step is going to be the signal that is going to be sent to the rest of the cell. There are a couple different pathways that you may see this happening for the EGFR that it performs, and that is the RAS-RAF-MEK-ERK pathway, which actually sends signals to the outside of the nucleus to signal cell differentiation as well as cell division. And then there's also the Pi3K ACK-mTOR pathway, and that one actually is going to stimulate more cell growth as well as control the cell's metabolism. So, attachment, dimerization, phosphorylation, and then finally signaling. Let's back up just really quickly. When we were talking about the phosphorylation, that actually happens in two different phases. The first one can be trans, where we can pass those phosphates from one enzyme to another and attach those. Also, there's additional tyrosines that are next to the membrane, and those also can be phosphorylated. So not just the enzyme itself, but also some neighboring tyrosines can also continue to be phosphorylated. Now, let's talk about this particular enzyme and cancer, okay? We know that cancer is the uncontrolled replication of cells. And so some cancer cells actually have a increased number of the EGFR receptors on the surface of the cancer cell. And so because of that, they definitely want to continue to divide and grow and make more of themselves, like cancer cells do. To try to inhibit that, we as scientists have come up with some inhibition ideas, because if we can inhibit the EGFR pathway from occurring, then we can stop the cancer cells from multiplying and growing, okay? Also, before we continue, I also want to throw in this little tidbit of information. The EGFR family also is very closely related to the HER family. And the HER family, HERs 1 through 4, there are actually 4 different enzymes, are extremely significant when it comes to breast cancers. I see commercials all the time on TV that talk about this medicine is effective for HER2 positive breast cancer and things like that. And so just keep in mind, some of the things that we're talking about with the EGFR also apply to HER as its own enzyme, okay? So there are 2 different ways that we can try to inhibit the EGFR pathway from occurring. The first one is that you completely stop the binding of a receptor. So if there's a way that we can issue or create some sort of drug that binds the receptor and prevents the EGF from binding, then that is going to stop the pathway from occurring. It's going to stop cancer cells from growing, dividing, and living, okay? So that's the first way that we can stop that. The second way that we can stop that is by attaching some sort of inhibitor to the enzyme itself. And if we perform that, of course, that's going to change the conformation of the enzyme and potentially stop it from working. Again, not going to send signals for the cancer cells to grow and divide. So there are several drugs that can be used to prevent and form conformational changes in the EGFR. And another thing is, not only does it change the individual enzymes itself, but it also prevents them from creating the dimer that they need to be able to send the signal. So if you can change the conformation enough that they can't dimerize together, then that is the way to block the pathway, okay? And so there are a couple different medicines here, cetuximab and panitumumab. Those have been effective for colon cancer, and colon cancer is something that has been affected by the EGFR protein itself. And there are also a couple different medicines here that will inhibit HER2 binding and dimerization as well. And that's trastuzumab and pertuzumab, okay? Those 2 medications block the HER2 binding and dimerization that can be used in the areas of metastatic breast cancer as well as some gastric cancers. So the first way, change the conformation, change the dimerization. The second way is let's block the receptor. And so to block the receptor, we use things that are called small molecules that are medicine induced. And there's so many of these small molecules that can be administered that if they bind that receptor site, we block the enzyme from working. And so some examples of that is, and there's several here, osimertinib, erlotinib, gefitinib, and afatinib, okay? Some of those are effective against metastatic lung adenocarcinoma, as well as lapatinib, which is used for metastatic HER2 positive breast cancer. There's also some different medications that are listed here for acute myeloid leukemia, as well as solid tumors that contain the TRK gene, as well as some mutated gastrointestinal stromal tumors that can be affected by small molecule inhibitors that block the receptor and therefore block the pathway. Now, there is the second family of receptors that we are going to talk about, and how those can be inhibited to block the cell division pathway, is the VEGFR family. And that stands for the vascular endothelial growth factor receptor. The VEGFR family, again, just is another type of tyrosine kinase. So we're looking at similar method or mode of action. But this particular one is actually responsible for the growth of new blood as well as lymphatic vessels. It plays a huge part in embryonic development initially, and is expressed as in the embryo, but it also is still relevant for adults because we are always growing new blood vessels, we are always growing new lymphatic vessels, and that's something that continually is happening in the body. Now, for this particular enzyme, it again acts as a dimer, okay? So we get attachment of the VEGF, which is your vascular endothelial growth factor, okay? We're going to get attachment of that, and then that is going to cause dimerization between two of the enzymes, that they are then going to autophosphorylate back and forth from one to another, and that is going to trigger the pathways of the RAS-RAF-MEK-ERK, which is going to signal some endothelial cell proliferation, as well as the Pi3K ACT, which aids in cell survival, and the PLC gamma PKC pathway, which assists in signaling for vascular permeability. Now, one of the ways that we can use the VEGF receptor is we're using it as a way to starve out the cancer cells. Cancer cells, especially tumors, need their own blood supply, and they have to live. They need oxygen, etc. They are cells, so they need those normal things that cells need: food, oxygen, water, etc. Okay? And so what we have done is come up with two different ways to try to inhibit the VEGF receptor pathway from taking place. Very similar to what we have seen before. So the first one is using those monoclonal antibodies, a medicine called bevacizumab. In this case, it's going to bind the VEGF ligand, and it's going to block the activation. Okay, so we're going to stop that dimerization, which stops the signal. This type of medication is used to target metastatic colorectal cancers, as well as non-small cell lung cancers, renal cell carcinomas, ovarian, cervical, and glioblastoma multiforme cancer, which is actually a specific type of brain tumor. In addition to the monoclonal antibodies, we also have developed some medications that inhibit this pathway that are small molecule. Satininib, sorafenib, pazopanib, all of those can inhibit the VEGFR kinase activity, and those are effective in renal cell carcinomas, gastrointestinal stromal tumors, pancreatic neuroendocrine tumors, thyroid cancer, and soft tissue sarcomas. So actually what happens is this is the name of one of the medicines, but it can actually come in here and it will block the VGF from binding. And when it does that, if you notice, we are going to essentially starve out those tumor cells. If the tumor cells cannot get more blood supply as it gets larger, it needs more blood, more food, more oxygen, etc. If we can starve out that cancer cell by blocking its blood supply, then that is one way to kill or slow the growth of the tumor. Now, there are several different members of the tyrosine kinase family. I have only talked about 2 of those today just due to time constraints as well as depth. But a lot of these different tyrosine kinase family members work in similar fashion. Again, we have that extracellular space. We have the alpha helices, the one alpha helix, that runs through the membrane. And then we have the inside portion of the enzyme that through dimerization and phosphorylation can actually transfer and signal messages to the inside of the cell, including the nucleus, as well as other chemical components. So there are about 90 of those tyrosine kinases that are receptors in humans. Some of the other families that act similarly as well as have similar drugs that can inhibit cancers and that we're working on currently in the scientific community to try to battle cancers are things like the PDGFR family, FGFR family. There's also a huge study and a lot of information going on the insulin receptor family, which that is a tyrosine kinase as well, that has to do with insulin binding. There's also the Ty family, the EPH receptor family, and the RET family. So lots of different opportunities here to create new drugs to affect this one family that will slow growth, cell signaling for growth, and division. Now, in summary, tyrosine kinases are a huge family. There's about 90 different ones. 60 are receptor ones found in humans, okay? I just kind of honed in on a couple of them today, just due to time constraints, but they help with signaling pathways for the cell. These receptor tyrosine kinases receive information from the extracellular environment through ligand detachment. Could be hormones, could be a drug, could be ions, could be lots of different things, okay? And through those ligand detachments, there's a conformational change in the enzyme that causes the enzyme to become activated. There's phosphorylation as well as dimerization, etc., that then is going to initiate a specific signal pathway into the intercellular space. These pathways in particular for the tyrosine kinases are focused on cell growth, development, as well as different types of tissue repair. Cancer cells some of them have a plethora of receptor tyrosine kinases, and we can use that to our advantage to try to block the cancer cells from growing and dividing if we can take advantage of the enzyme itself as well as its receptor component. Using medication, we can stop cell division, halting cancer cell growth, and proliferation of the cell. And here are the references that were used for the presentation.